right, perfect timing. I think our second speaker is here, so we will go ahead and welcome everyone again. Angela, did you want to just check your sound or anything like that before we start?
Yeah, can you hear me okay?
Sounds
Is that
good.
good? Awesome.
Thank you. So yes, welcome everyone to another edition of the Black and Comp Bio series. My name is Janae, and I'm your host for today. Today we will have Dr. Angela Brooks and Alexis Thornton joining us from UC Santa Cruz. And the Brooks Lab focuses on the study of somatic mutations that cause changes to the transcriptome, particularly through mRNA splicing. They aim to gain a better understanding of how alternative splicing is regulated and the functional consequences of splicing dysregulation through the study of cancer genome alterations. They're developing computational approaches to analyze genome and transcriptome sequencing data and developing high-throughput experimental approaches to characterize the functional impact of cancer variants. Much of their work has been in collaboration with large national and international consortia, including modENCODE, the Cancer Genome Atlas, and the International Cancer Genome Consortium. So welcome the Brooks Lab, and go ahead and take it away.
Thanks so much, Janae, for that introduction. And, thanks so much to you and, the other members of this group to make this really awesome community. Really happy to talk about, I'm going to start off just talking kind of broadly about the way our lab thinks about understanding cancer associated variants, and then I'll hand it off to Alexis. And I will say, I don't think I've ever mentioned specifically to Alexis, I forget if I have, but as I've gone through my own training, I think Alexis is probably the first Black woman computational biologist that I worked with. So, having this kind of network is really amazing. So it's good to see everyone here. So let me start sharing my screen. Okay. All right. Hopefully you can see my slides. So thanks again, everyone who's here. So I'm Angela Brooks. I'm assistant professor at UC Santa Cruz. I've been here for about almost five and a half years now. And the title of my part of the talk is High Throughput and Full Length Characterization of Transcripts and Their Function. So the way to frame the work of my lab, we're interested in cancer precision medicine. And so when an individual unfortunately has cancer, we want to be able to take a biopsy of that sample sequence and look at the types of mutations that exist. And maybe that'll help to inform what type of therapy will work best for them. But one of the challenges with this is, as we've been sequencing more and more cancer genomes, we're seeing millions of these mutations. And so really understanding which of these mutations are actually contributing to cancer and which ones might be passenger mutations is becoming even more challenging. And also, which of these mutations, even if we know how they contribute to cancer, how can we use that to inform therapeutic decisions? And so the typical way that we think about how to identify which mutations are actually contributing to cancer versus passengers is to do large-scale sequencing. So for example, say in lung cancer, you have a large cohort of say 500 tumors that you sequence. Then you look for commonly mutated genes. So for example, KRAS is one of the most commonly mutated genes in lung cancer, followed by, let's say, EGFR is another gene. But most mutations are actually very rare. So, KRAS, EGFR are sort of the exception rather than the rule. So most cancer genomes, these are very rare events that are occurring. So the way to understand, or the way our lab aims to try to get a better understanding of these mutations is to also look at the associated RNAs that are present in those samples. And so one idea is you have, say, a cohort of healthy donors or tumors that are wild type for a mutation, and then samples that have a tumor mutation, and then look for what are the associated RNAs to gain a better understanding um, of the functional impact of that mutation. So the types of mutations that our lab studies are those that affect splicing. So my graduate work was to understand splicing regulation kind of more generally, and I sort of applied that to cancer in my postdoc and now in my own lab. Basically, when I started my postdoc, this is when many cancer genome studies had started coming out to show that splicing factors are very recurrently mutated in cancer. So this is a figure from a review showing in many different cancer types the frequency of mutations in these 4 core spliceosome factors. And one of them is a gene called SF3B1, and this is a gene that my lab has followed up with, and largely in collaboration with Catherine Wu. And so SF3B1 is one of the most recurrently mutated gene in chronic lymphocytic leukemia, which is one of the tumor types that we study. And so it's just as recurrent as say P53. And so a lot of the work in my postdoc and some of the work in my own lab is to ask what are the RNAs that are associated with SF3B1 mutation, and understand what are the maybe splice variants or altered RNAs associated with this mutation. We can maybe better understand its role in cancer. So what do we know so far about SF3B1? It's associated with the branch point, which is a, let me get, see, I think I can, oops, sorry about that. I was trying to get my pointer, but I'll just use the arrow for now. So SF3B1 is a protein that associates with the branch point sequence. So this is important in the first step of splicing. So I'm blazing over more details of that, but essentially what we know is in wild type case that SF3B1 can recognize a normal branch point and you get sort of normal splicing. But in the presence of one of these mutations that was observed in cancer samples, again, in particular in chronic lymphocytic leukemia, there's an altered or a different branch point that's selected, and that results in what we call cryptic 3' splice site usage, which just means you have a different 3' end of the intron that's selected, and so you get a different aberrant splicing product. So so we've identified the mechanism of how SF3B1 affects splicing via using short read RNA-seq data. Briefly, this is just a sequencing method where you're taking cDNA and then sequencing bits of it, and you can get it from primary samples. And so one question then is, were we seeing these alternative 3' splice site events that are associated with SF3B1 mutation, but we don't quite understand what's the functional product of these splicing events. So in short read methods, we're sort of looking at just a local event. So is this 3' splice site used, or is it skipped in another isoform? But what we know about actual mRNA transcripts is they're very long. These splicing events are typically examined, especially with short read data, at a localized region. And so knowing the full isoform will help us better understand what the function of those transcripts are, because we'll get the full sequence. So for example, maybe that alternative splicing event, when used in the context of a full transcript that is determined to be unproductive, meaning not coding for protein. This is usually identified by transcripts that have a stop codon 55 nucleotides upstream of a splice junction. You might also have different protein isoforms that are linked to an alternative splicing event. So if you're only looking at one particular region, you're missing other information of the transcript. And so a lot of work in our lab is to use long read technologies. We're primarily using nanopore sequencing to sequence full length transcripts. So briefly, the way this works is you take a nucleic acid, say RNA or DNA, and you run it through a pore, and you get a current signal as the nucleic acid goes through. The certain sequences sort of block a current, and you get changes in that current as it processes through. It's read, the signal corresponds to a chimer that has blocked the pore. And as it processes, you get overlapping chimers, and then you end up with a consensus sequence at the end. And the benefit of this technology is you can sequence very long transcripts. So this is kind of a little graphic of some of the pros and cons of using short read sequencing versus, say, long read or nanopore sequencing. So for short read sequencing, what's great about it still is highly accurate. You get a lot of reads from it, and there's a lot of established computational methods to analyze this data. But it has been shown that it's very difficult to infer full-length transcripts from these short reads. In nanopore, you have long reads. Although this is improving different methods, but particularly for nanopore, it tends to have lower sequencing accuracy. Again, things are improving, but it's still not the level of Illumina for most standard approaches. You get lower read depth compared to, again, for the same amount of money, you get lower read depth from nanopore sequencing. And then there are fewer established computational tools. And so our lab is kind of focused more on, what kind of computational tools can be developed to analyze this data. So we applied nanopore sequencing to ask this question about what are SF3B1 associated transcripts. And so this is work from a grad student, Allison Tang. So we took 3 normal B cell samples, 3 CLL samples that were wild type for SF3B1, and 3 that had a hotspot mutation in SF3B1. We did created cDNA and then did nanopore sequencing. And so the big question again, how do you analyze the data? So we built this tool we called FLAIR, called full-length alternative isoform analysis of an RNA, and it's kind of a full-featured product, I guess you could say. It does transcript assembly from the long reads. It does quantification. It will predict whether the transcript's productive or not. I mentioned that 55 nucleotide rule. It'll look for differential expression given a particular data set, and it'll also look for localized differential splicing events. And so using long reads, we were able to get a full context of these sort of local events, as I mentioned. So here's an example of an alternate 3' splice site in this gene ERGIC3 that had been identified with short read data. So this is just quantification of the relative usage of that 3' splice site. So in the wild type CLLs, you almost always use this more distal splice site, but with the SF3B1 mutation, you're using this more proximal site. With the long read data, we're able then to get the full context of the transcript and where that splice site exists. So what I'm showing in blue are those full-length isoforms that use the distal site. And what I'm showing in green are all the full-length isoforms that are using the more proximal site. So you can see that beyond just this local event, there's a lot of additional splicing complexity that are associated with those splice sites. And in addition, we can do differential isoform analysis, and that allows us to be able to look at coupling of different types of alternative splicing. So here's a case where you have intron retention kind of in the beginning of a transcript, and then alternative 3' splice sites kind of towards the end of the transcript. And since you can quantify the full length, you can actually quantify how those different splicing events are coupled together, and see that there is a association of certain intron retention events associated with certain 3' splice sites. So I really just breezed through that paper. It's published. I just wanted to give a flavor of kind of the long read analysis that we've been developing in the lab. And we developed this tool FLAIR where improving on it. We have new projects to add on to this tool. Um, what we showed is we can use long read data to recapitulate a lot of the findings that you can find with short read data. And what long read is allowing us is to see a more complete picture of the transcriptome. So going back to this question, what are the functional outcomes of these spliced products? We're still interested in this question. As I mentioned, computationally, we can kind of predict what's an unproductive versus a protein coding isoform. But even when you have these protein coding isoforms, you may have only subtle differences in the sequence, but how that changes the gene function is really unclear. And so to give a flavor of how challenging this might be, this is an example from our 2016 paper where we're characterizing SF3B1 just using short read data. And so in that study, again, this was in collaboration with Kathy Wu and her lab, we found this really interesting. So when we're looking at the data, we found this really interesting splicing event in a gene called DVL2, which is disheveled, which is a gene that was really well known in the Wnt pathway. And we could clearly see, and this is kind of recoverage of a sample that is wild type for SF3B1 and one that's mutated for SF3B1. You can really see this drop in coverage that's associated with an altered splice site usage in this gene. And so we wanted to know, how does this splicing change affect disheveled function? So it took 3 years and 2 postdocs and some technicians to actually figure out that this isoform change is actually affecting the Notch pathway. Again, disheveled was known to be part of the Wnt pathway, and so there was a lot of follow-up about that. But very long story short, we were able to see this effect on Notch, and that this this altered splice variant of disheveled that's caused by the SF3B1 mutation actually causes an activation of the Notch pathway. So that brings us, this is kind of an introduction to Alexis's project, which is in thinking about how do we more rapidly characterize genetic variants. In particular, we're interested in cancer-associated variants, in particular splice variants. How can we more rapidly functionally characterize these? So we have a ton of variants we're identifying from sequencing data, but a lot of times we use these gene-specific experimental assays. So for example, the disheveled gene, known to be in the Wnt pathway, check Wnt pathway, assays, but nothing kind of came out of that. So we really need other approaches to think about how do we, in a very high throughput way, characterize many variants at a time so that we can more rapidly understand their function. So Alexis will give you more details about that project, but I want to just close and thank the people involved in the SF3B1 and a lot of our nanopore sequencing projects, including Allison and Cameron in particular. I wanted to give a small plug to our graduate program at UC Santa Cruz, the PBSC program. Unfortunately, we just had our application and interview process, but if you're interested in the future, the next applications will be due in December of this year. This is a picture of where you could do research, or if you are not staying at home elsewhere. We're still largely stay at home, especially our computational folks, but we live in a beautiful place near the water, also with a lot of trees and nature. And then finally, before I hand it over to Alexis, if anyone is interested in long read transcriptome analyses or developing methods for analysis, I'm co-leading an effort called the Long Read RNA-seq Genome Annotation Assessment Project. Here's a website, and we're generating a whole bunch of data with different long read platforms, and we're going to be evaluating both the platforms but also computational methods for analysis. So with that, I'll stop so I can hand it over to Alexis. And then I think we can probably just take questions at the very end because I want to give Alexis time. for her talk.
All right, hopefully you can see my screen. I'm Alexis, yeah, I'm a 4th year grad student in Angela's lab, and I'll be talking about our new tool called EVIP2, and a lot of this work is on bioRxiv, but there's some new stuff in this presentation. So like Angela just said, there's been millions of mutations identified by these large consortiums. But for most of those mutations, we don't know their functional impact. And gene-specific experimental assays are often used, but they're pretty slow and time-consuming. But expression profiling methods can profile the function of many variants at once. And that is the motivation behind EVIP, which is to be able to characterize variants in a more high throughput way. The EVIP approach is to compare expression changes upon overexpression of different gene variants. Wild type and mutated open reading frame libraries are overexpressed into cells using cell line transduction. Then the gene expression measurements are used with the EVIP algorithm to predict the function of the mutation. And that function could be loss of function, gain of function, change of function, or neutral. And the idea is that by comparing the gene expression changes that are induced by wild type and mutated alleles, you can get the functional insight without requiring prior knowledge of the gene's function. And in the original paper from Angela and Alice Berger that presented EVIP, they used 8 replicates per condition. And having 8 replicates was made possible by using the L1000 assay, which only measures the abundance of 1,000 genes, and it's kind of a cheaper way to do it. And it was created for these types of high throughput expression profile applications like EVIP. And so these are the 3 main comparisons that the EVIP algorithm uses. The first is the self-correlation among those 8 wild type replicates, then the self-correlation among the 8 variant replicates, and then comparing the wild type replicates to the variant replicates. And these heat maps represent those correlations. And so here's a simplified version of the EVIP algorithm that eventually predicts if a mutation is neutral, change of function, loss of function, or gain of function. At the root of the tree, we ask if the wild type signature is the same as the variant signature. And if it is, then the variant is neutral. So you can see in that last heat map, when comparing the wild type replicates to the variant replicates, that correlation is high. But in these other non-neutral mutation calls, that correlation is low. And so when the wild type and mutant signatures are different, we then compare the strength of the signal by looking at the self-correlation of the wild type replicates and compared to the self-correlation of the variant replicates. So now we're comparing these first 2 heat maps. And when they have a similar correlation, we consider it to be change of function, but when there's some type of difference or directionality in that change, it's a loss of function or gain of function. So it's gain of function when the mutant self-correlation is greater than the wild type, and it's loss of function when the mutant self-correlation is less than the wild type. And so to also explain the approach in another way, these are transcriptome-centric definitions of the EVIP calls, which may differ from classical genetics terms. And so neutral mutations effect on the gene expression is the same as wild type. Gain of function mutations have a strong effect compared to wild type. Change of function mutations have a different effect compared to wild type, but there's not enough significance for loss of function or gain of function calls. And with the loss of function mutation, its overexpression causes a mild effect. And so in that original paper with the EVIP method, they profiled 110 lung cancer mutations, which are represented here in what we call a sparkler plot. The x-axis represents the significance, where the y-axis represents the impact direction, where a positive impact direction score means it's gain of function, and a negative means loss of function. And the ones in between are our gain of function, or change of function calls. And when dividing all of these mutations into the type of gene it occurs in, we see oncogenes are enriched for gain of function and change of function calls, and the tumor suppressor genes are enriched for loss of function. And this is consistent with what we would generally expect. And so I've been working on EVIP2, which is a more automated version of that original EVIP software. And the main element is that it can now use RNA-seq data as input. And so this is an overview of that EVIP2 pipeline, where we use Callisto gene expression data and process it with Z-scores and Spearman similarity matrix, which is the input to the EVIP algorithm, which then determines the functional prediction. And a new functionality I created was that we can now also predict the functional impact for each variant on the pathway level. And this pathway level analysis was made possible by using RNA-seq, which gives a more complete view of the transcriptome than the L1000 assay would. And so the connectivity score is a measure of correlation that is specific to the L1000 data that was used in that original version of EVIP. So moving forward with the RNA-seq data, EVIP2 uses the Spearman rank correlation. And so when rerunning the 110 variants with EVIP2 and the Spearman rank correlation, we see a high correlation to the original L1000 results. And this gave us confidence in moving forward with the Spearman rank as our correlation metric. And so we thought that these two variants would be a good application to use EVIP2 with. RNF43 is a negative regulator of Wnt signaling. It has these two recurrent mutations that cause frame shifts at position 117 and 659. And this figure shows colorectal cancers, but RNF43 is also commonly mutated in gastric and endometrial cancers. And because this is an interesting case where there's one frame shift early on in the gene and another one later in the gene, we thought that this would be a good use case for applying EVIP2 to investigate if these two variants are causing the same impact on gene function or some type of different impact. And so it's usually assumed that truncating mutations would cause a loss of function, which is what we saw with this earlier frame shift, but the later frame shift was actually a change of function, which was really interesting because you may expect it to be loss of function, but maybe also to be neutral since it was so close to the end of the gene. So the change of function call was really exciting. And we can see with these overall results, we are predicting that the variants are having a different impact on the gene's function. So with the pathway analysis, we then dove into what those pathway level impacts could be for each mutation. So EVIP2 uses two different gene lists to generate pathway level predictions. We compare the control replicates to the wild type. And in this case, the control was GFP. And we also compare the control to the mutant. And then we compare the differentially expressed genes that happen in those two comparisons against each other. So the mutation specific genes are the genes that are specific to the control versus mutant comparison, and the wild type specific are specific to the control versus wild type comparison. And I think of these mutation specific genes as the genes that would represent a new function that the mutation causes, where the wild type specific genes represent a function that the mutation has lost from the wild type. So based on that, we expect the loss of function to have more wild type specific genes, and we expect the gain of function to have more mutation specific genes. And that is consistent with what we saw, where the early frame shift has more wild type specific genes, and the change of function, which had a trend towards gain of function, has more mutation specific genes. And we then run pathway analysis on each of these gene lists separately. So for the loss of function, we focus on these wild type specific genes, where for the change of function, we focus on these mutation specific genes. And these are our pathway results using the 50 hallmark pathways from MSigDB. The loss of function had mostly pathways with a negative impact direction score, except for the KRAS signaling up pathway, which represents genes that are upregulated by KRAS activation. We found that one to be gain of function. And that gain of function call was really unexpected in this wild type specific gene set. So that was a really interesting result for us. And then in the change of function variant, most of the calls had a positive impact. And the KRAS signaling up and down were also gain of function in the 659 frameshift mutation. So from these results, we can see both of them affect the KRAS pathway, but the change of function variant is also affecting other pathways as well. And so for experimental validation, we then focused on these 6 pathways that have the most significant mutation impact p-values. And so this work was done by Lashan Fong, who is a postdoc in the Giannakis lab. And this reporter assay measures the expression in the control, wild type, the earlier frameshift and the later frameshift. ERK and JNK are part of the MAPK RAS signaling pathways. So we use those to evaluate our KRAS pathway predictions. And in both of them, we see activation in both variants, but no activation in the control or the wild type. So that validates that something new is happening in those mutations. And we also see the change of function variant has higher activation than the loss of function one. And the change of function variant also is activating hypoxia and NF-kappa B, which are consistent with the predictions. So all 4 of the pathways that were activated here were all found to be impactful with EVIP. We did have a false positive for P53, where we predicted it to be gain of function, but there's no difference among the conditions. And so I think the reason for that was that pathway mutation-specific gene list had 40% overlap with these other pathways that were activated. So just going forward, we will take that kind of gene overlap into consideration. And then Lashan also used Western blots to further validate the activation in those pathways. EMT was an additional pathway that she verified that wasn't in that original reporter assay. So where in the assay, the loss of function had ERK and JNK activation, we don't see that as much in the Western blots, maybe a little bit for JNK, but so we think maybe the weaker transcriptional change that's caused by the earlier frameshift was not strong enough to result in pathway activation in the Westerns. So this is the RAS MAPK signaling cascade that we're finding the variants are affecting. And it's commonly altered in colorectal cancers. Mutations in KRAS and BRAF are common, and they result in constitutive activation of this MAPK signaling cascade, resulting in uninhibited cellular proliferation and tumor growth. And with analysis using patient data from TCGA, we found that RNF43 truncating variants have a similar expression profile to these BRAF mutations, which suggests that those patient samples with the RNF43 variants are also activating this MAPK signaling cascade. So this is that analysis that found that connection between the BRAF and the RNF43 variants in the patient data. We use a tool called LUR, which uses known driver events as bait to then catch events that have similar expression profiles. So this found that those BRAF missense patients found 42 samples with similar profiles. And so again, the BRAF missense mutations are known to activate the RAS MAPK pathway. So this is a good sign for us that it was consistent with our EVIP predictions and also with what we saw in the experimental results. And so this oncoprint basically shows where each column is a different patient. And so there are samples that have both the BRAF and RNF43 variants, but there are 42 that don't have that, the BRAF variant. So that suggests that in these patients, they are having that expression profile independently of the BRAF event. And so here in this box plot, I'm comparing the LUR classifier scores in samples with different BRAF and RNF43 status. The LUR classifier scores represent the BRAF mutant signature. So the samples that are wild type for both have low scores, where the samples that have both variants have the highest scores. So the key takeaway is that even in samples without the BRAF variant, there is still high classifier scores. And also the samples that have both are significantly higher than the ones that only have the BRAF variant. So maybe together there's some kind of combined effect. And so LUR actually uses all of the RNF43 truncating variants together, not just the two that we looked at with EVIP. So I then looked to see which variants it identified. And so it identified all 6 of the 117 frame shifts and all 31 of the 659 variants that were present in the data. So then to remove any ambiguity, I took out samples with any co-occurring BRAF, and I also took out samples that have multiple RNF43 mutations. And this left 6 variants with the 659 frame shift and 1 with that earlier frame shift at position 117. And then when looking at the classifier scores in these remaining variants, we see that our change of function mutant has stronger classifier scores, meaning that they're more BRAF-like than the earlier frame shift, and also more than these other ones that are less recurrent. And so this stronger classifier scores is consistent with our results of that variant being a change of function and having a stronger impact than the loss of function one. So then just to verify that there is activation in those genes, I use a 10 gene MAPK pathway activity score signature with a few Wnt genes and MLH1, which is known to be downregulated in these RNF43 variants. And we see that the variants mostly cluster together regardless of their BRAF status. And we see activation of MAPK genes, especially in EPHA4, DUSP4, and ETV5. So we do see evidence of some of these MAPK genes being activated. I also benchmarked EVIP2 against two gene set enrichment analysis tools. And so to account for all the different inputs and options, I ran 80 different variations of GSEA and 12 different variations of FGSEA. Which is why there's so many different points for these other tools. But with EVIP2, we only ever used the default parameters. So that's why there's only 1 point for EVIP2. And so none of the other tools reached the 100% sensitivity that we did. And the ones that had a lower false positivity rate didn't have a very high sensitivity. So we think that it's possible when trying to predict what a mutation's impact is, EVIP2 may be a better choice than traditional gene set enrichment analysis methods. And lastly, I just want to mention this other publication that came out at the end of 2019 that suggested that the RNF43 G659 frame shift is a passenger mutation, which is not consistent with what we see. And so since RNF43 isn't very well characterized, but it is known to negatively regulate the Wnt pathway, they just focused on the Wnt pathway and they totally missed how it affects these other unexpected pathways. And I think that highlights how useful our EVIP approach is when the gene function isn't well studied. Yeah. And so just to conclude, EVIP2 now works with RNA-seq data and can do pathway analysis. Our results were validated in cell line experiments and was also consistent with patient data. And in the future, because the Brooks lab is mostly a splicing lab, the next steps for this project would include using EVIP to characterize cancer-associated splice events by comparing dominant wild-type isoforms versus maybe a cancer-specific splicing isoform. Yeah, and I want to thank everyone in the Brooks lab, especially Angela and everyone else who worked on this EVIP2 paper from the Giannakis lab and the Berger lab and the Stewart lab. And this is our picture from, I guess, 2019 summer bonfire that we do on the beach in Santa Cruz, which is always lots of fun, though we didn't get to do one last year. I look forward to the next one, and that's it.
Thank you both so much for that. It was a really great presentation. I will let the audience go forward with any questions you might have. Feel free to put them in the chat. It may be better for you to just unmute and ask.
You can also feel free to ask questions about like career path or, you know, not just about the science or if you want.
I have a couple questions, if you don't mind me just asking up front. Very nice talk and very interesting data, and thank you both for sharing about your lab, Angela, and then about your research, Alexis. So I was curious about the, I guess, like this discussion of the change of function that you were identifying using the EVIP tool. And to kind of, I guess, look more deeper into that, have you thought about doing any kind of time course sort of experiments where you would look at how, if you perturb the system, like how maybe the signaling might change for some of these genes that you're seeing that have these change of function outcomes, so that you could actually see what is the change. Is it this this gene that is normally part of the MAPK pathway is signaling with something else or something else? Or have you thought about other experiments that you could do to actually identify what these change of functions might be?
I haven't thought of any experiments. I mostly do the bioinformatics side, but yeah, I'm not sure. I don't know. I know this data is from the Giannakis lab, and they're doing a lot of other experiments on these specific variants, but I don't know if they're doing any time course experiments.
Yeah, would be interesting. Oh, yes.
Oh, I was going to say another context that we tried in the original EVIP paper is different cellular contexts. And so for the study Alexis presented, it was only done in one cell line. And definitely what we saw in our original paper is that if you have different cell line contexts, that the function can change because, you know, maybe there's, you know, different other factors that are expressed in different conditions. So we haven't done sort of time course experiments, but definitely different cellular contexts, and have noticed that there are differences in the predicted impact.
Okay, yeah, it's a hard thing to get at. I tried doing some of that work early in my grad studies, and it's it's very difficult. And the other question I had was if you had any thoughts about incorporating any of the new single cell sequencing technologies to look at the heterogeneity of these splice site events and the effects on the gene expression, like ATAC-seq and things like that. Whether it's part of the Brooks lab or future directions of this sort of work, Alexis. So
I think there is a single cell approach. Angela, was it called, is it perturb-seq? Or it's some continuation similar to EVIP that uses single cell data, but it doesn't use, it's not for splicing.
Yeah, definitely. Yeah, I know that there were efforts. So when I originally worked on this approach, it was at the Broad, and I remember that there was some efforts to do similar approaches using single cells so that you could get, you know, thousands and thousands of expression profiles associated with the variant. So definitely, you know, I think we'd hope that this EVIP algorithm could then just plug into any expression data. So if it was single cell gene expression, I mean, there might be some caveats with the sort of other caveats of single cell, but in theory, you know, using just any expression signature data, in theory, could be used with the EVIP approach.
Thank you.
Any other questions?
Hello. I also have a question. Very nice talks about. I have been following your work, and I think it's very interesting. I was wondering in your project, Alexis, whether the interpretability of certain variants can be kind of influenced when you find like two different variants in a patient, like if a combination of different variants then change the interpretation of if you have loss if you have loss of function or gain of function in particular pathways. Yeah, I'm more interested on this side of the combination of mutations. I think that's very interesting.
Yeah, it is really interesting. We actually, I recently ran some data on EVIP2 that's related to that of a patient or cell lines that have two different mutations. So that is something that we are trying. But for validating in like the patient data, like I was saying, like kind of does make it complicated because you don't know exactly what mutation is causing the transcriptional profile that we're looking at. And I don't know if there's any way around that.
Thank you. And I have another question more in the splicing side, whether and now with this long read technology, many people are saying that sometimes it's not very quantitative, or like it that would be kind of like the major limitant to keep using it, to let the short reads away. But I see that your approach kind of gives a quantitative difference on the isoforms. What is exactly what the developments that have led to make this technology more quantitative, and what do you think it will be kind of the future that do you think it will at some point replace completely the short read sequencing, or what is your expectation on this?
Yeah, that's great. Great questions. It's definitely quantitative in terms of, if you look at replicate reproducibility of replicate sequencing, or if you comparing with, say, spike-ins or some other type of benchmarks, I think there's many papers that have shown that it is quite quantitative. I think one of the main limitations of why it's not maybe as quantitative as, say, short read sequencing is just the depth of sequencing. So you're not, because you have sort of shallow counts, you're not quite getting like the most dynamic range, or it's harder, especially for lowly expressed transcripts, to get a better sequence quantification. So I would say that there's definitely interest and effort to sort of take long read sequencing as, replacing short read sequencing. There's still, again, the depth issue, especially the same a fixed cost. There's also a limitation that we've been finding, others have found, that it's not quite long enough. So for really long transcripts, especially for methods that are using PCR-based approaches to convert to cDNA, or there is some kind of limit to how long you can get. So then you get a bias in your representation of the transcriptome. So those are some kind of remaining issues. But definitely this LR-GASP consortium that I mentioned in my slides, and I'll actually like post a link in the chat if anyone's interested in in these approaches and improving quantification and transcript detection with long reads. There's definitely a large community effort now. We're bringing in experts of long read technologies in different spaces to try to tackle this problem. And I think it is a goal of our group to try to bring long reads to kind of replace short reads, at least for the application of transcriptomics.
We do have one question in the chat from Winfred. Very nice presentations. Just a quick one. A majority of variants identified via GWAS are located in non-coding regions. Will you consider eQTLs and epigenetics in the future, since I believe they may affect the gene pathways?
Yeah, that's a great question. Yeah, we haven't. I guess our next direct projects won't involve sort of non-coding region effects unless it affects splicing, because we're sort of more interested in the downstream effects of splicing. But that's a great question, and definitely really important, since a lot of disease-associated variants are non-coding regions. So, but I do know there are many other groups working on this problem as well, like high throughput reporter assays to try to functionally characterize these non-coding regions. But at least our immediate projects aren't going to tackle this yet.
I guess my last question for the both of you is just, what's the most exciting part about like working in this sector of like RNA biology and computational approaches to understanding it? Yeah, just as someone who's also in this same field.
Maybe I'll let Alexis go first.
I guess the most exciting part is just like how much data there is available. I think it's really cool to be able to like have a question and find like data and experiments that have already been run that you can help use to answer your question.
I think for me, I've really enjoyed the RNA community. So I started as a graduate student. I was more in kind of basic RNA biology space, and even now, I have colleagues that I knew as a graduate student in the RNA world, and it's a really warm and open community. So I've really enjoyed working in RNA computational biology.
Agreed. Well, thank you everyone for coming today. Feel free to reach out to them with any other questions you have. I'm going to put the link here to register for our next seminar. And for those who couldn't make it today, I'll try to have the video posted somewhere in the next few days, grad student pseudo time. So again, I appreciate you all for being here, and have a great day.
Thank you.